All right, so now we're going to talk about the last two fatty acid-based lipids. These both fall into this category of phospholipids. Phospholipids. We're going to see that's because they contain a phosphate ion. And those are glycerophospholipids and then sphingolipids, which are a pretty specialized class. Okay, so phospholipids, what makes them phospholipids is this guy right here, which is a phosphate. If it was by itself, it would be PO4 minus. In these molecules, much like what we've seen before, it forms what's called a phosphoester bond. Okay, so it's not quite our typical ester bonds that we're used to, but these are called phosphoesters, and so we can expect a carbon, well, and this should be a negative charge here. There's some sort of a carbon chain happening on either side of that phosphorus, oxygen, and then carbon bond. It then also has that negative charge. We're going to see that these groups right here in general are polar. They are charged groups. And that's going to have a big impact on how these lipids are different from the other two classes of fatty acid lipids that we talked about. Importantly, though, both of these do indeed contain fatty acids. For the glycerophospholipids, you again have that glycerol base, and there are two fatty acids attached. Okay, and that's where we're going to start off, talking about these glycerophospholipids. So again, you have your glycerol, right? That's that component. Two of those glycerols are now going to be replaced with our fatty acids, which again form those ester bonds. And then the other one is going to have, so those chains would be longer, right? Because they're fatty acids. The other one is now going to be replaced by that phosphate group, forming a phosphoester bond with what's called an amino alcohol. So then on the other end, we have our amino alcohol. Okay. The sphingolipids or sphingomyelins are kind of their own special thing insofar as they're not glycerol based. They don't have that same glycerol connectivity. They have what's called sphingosine and myelin, like myelin sheaths on a nerve. These are lipids that are abundant in nerve tissue as well as in the white matter in your brain. So they play a fairly specialized role. The glycerol component, as well as one fatty acid, we'll see this whole chunk is kind of unique to these sphingolipids or sphingomyelins, and it's called sphingosine. It does have one fatty acid component. And then again, because we're in the phospholipid section, it has that phosphoester bond to an amino alcohol. Okay, so these are these last two groups of lipids we're talking about that are fatty acid based. We still haven't gotten to steroids yet. Amino alcohols, what the heck are we talking about there? So these are the three amino alcohols that we're going to be talking about that are a part of both the glycerophospholipids as well as the sphingolipids. You can see that they all have, so this is the portion that would be connected, let's actually draw it here, connected to the phosphorus, all right, so this would be the phosphoester bond, P. And then it's connected to the either glycerol in the case of the glycerophospholipids or the sphingosine in the case of the sphingomyelins. The other key piece to point out here is they have these charged amino groups, charged amine groups rather. Okay, so they are incredibly polar because they have this net charge on them. All right, so there's the three that we're going to learn about: choline, serine, and ethanolamine. And these are also given on your constants page, okay? So this was the constants page that we had for chapter 17. It had all of our different fatty acids. The second portion of this page would be our 3 amino acids. And then look, you can see here's what sphingosine looks like. We'll talk about that here in a second when we get there. Okay, again, important feature, this positive charge, which makes these groups all very polar. All right, so if we wanted to sort of zoom in and look at what one of these glycerophospholipids would look like, we again have our glycerol backbone. Okay, we have, for the glycerol phospholipids, we have 2 units of fatty acid. And they could be anything from our constants page. Let's take a look here and see if we can identify what these fatty acids are. They have no double bonds, so they're saturated fatty acids. And they have 1 plus 14, so 15, 16 carbons. So these would be these palmitic acid components. All right, now we have one of these things that are unique to the phospholipids, this phosphoester bond here. And that is binding our amino alcohol, which is in this case choline. Again, notice that positive charge. Right, and so the 2 components, you have these non-polar fatty acid components, but then these polar amino alcohol components. And so this is another one that has the same palmitic acid fatty acid component, but now instead of having a choline amino alcohol component, has the ethanolamine amino alcohol component, okay? Still has that same features of glycine backbone, glycerol rather, backbone, phosphoesters, 2 fatty acids, and then just a different amino alcohol. Okay, so that's the anatomy of a glycerophospholipid. These are 2 famous ones, lecithin and cephalin. Both of them are also abundant in tissues, but also found in like egg yolk and yeast, other components like that. So they're fairly abundant. Okay, and kind of been saying this all along, but this is the really important aspect of these glycerophospholipids. The fact that one end of them is a polar end, which we call the polar head, that consists of that positively charged amino alcohol. And then we have a non-polar hydrocarbon, what we call the tail, and that is the non-polar region made up by, these are the fatty acid components, fatty acids. All right, so glycerophospholipids have one end of them is polar and one of them is non-polar. Okay, so often what you'll see is these glycerophospholipids drawn in such a way where one end has that polar end and the other end has that non-polar tail. So if we wanted to look specifically at a particular glycerophospholipid here, we have this amino alcohol head. Again, notice this phosphate group in here forming that phosphoester bond. And then we have the non-polar tail. Why is it drawn like that? Well, I mean, first of all, these 2 ends don't really want to interact with one another, right? Non-polar molecules don't like interacting with polar molecules and vice versa. So it makes sense that they would be separated in space. The other reason is because what am I trying to say? The place where we see these things the most are actually in membranes. So cell membranes are made up of these glycerophospholipids, and they look a little something like this. Okay, let's just zoom in. These are different like kind of transports of stuff going into a cell or out of a cell, whatever. We're not going to focus on that. Let's look at the actual cell membrane itself. Each one of these is a glycerophospholipid. And the green portion represents the polar head, and the orange portion represents the nonpolar tails. So membranes are called, another word for membranes are lipid bilayers, because what we see are these phospholipids where the tails, the nonpolar ends, are touching. And then the polar ends, the ones that are actually touching, you know, the intercellular fluid, right? I mean, cells are mostly water, so mostly polar solutions. So you have these polar heads separating, for example, the inside of the cell and the outside of the cell. And the membranes are made up of a ton of lipids stacked side by side by side by side, where their polar heads are all touching, their nonpolar tails are all touching, and then just as importantly, you actually have two of them stacked back to back, which is what forms the membrane of a cell. Okay, so cell membranes are made of these glycerophospholipids. So kind of crazy, right? That's why there's such a large class of protein, of lipid, because they're super key. I mean, in a sense, they're essential to how different cells are organized, right? We have certain cells are made for your liver, certain cells are made for the heart, certain cells are made for all your different body parts. How are they all kept separate? Through these glycerophospholipids that form these lipid bilayers, forming what is broadly called the cell membrane. Okay. Another important aspect of these glycerophospholipids, again, by virtue of the fact that they have these polar components and nonpolar components, is they're actually used to package other lipids, right? So other lipids that are, for example, triacylglycerols that have only nonpolar components. How do you go about moving those, right? Those aren't water soluble. So what ends up happening is they form what are called lipoproteins, which is essentially like a bubble. Okay, if we want to zoom in on here and see what's going on, this is a bubble where on the inside, this stuff here, these are all non-polar lipids. They could be cholesterols, so we'll talk about that when we get to steroids, or what we've talked about before, these triacylglycerols, but things that are strictly non-polar. And they're packaged in this bubble of glycerophospholipids, where on the outside of the bubble, oh, I used my green, gotta change colors. On the outside of the bubble are the polar ends, and on the inside of those bubbles are the nonpolar tails, right? So the polar heads are on the outside, which is why these things are water soluble. They can be carried through a cell, which is an aqueous environment, a very polar environment. But they contain all these nonpolar lipids because on the inner lining, again, if you kind of zoom in here, you can see the inner lining of this are all these nonpolar tails. So it's a way to keep both the outside polar environment happy, as well as the inside non-polar environment happy by creating these lipid protein spheres. They do have, I sort of glossed over it, they do have these membrane protein components kind of studded throughout. And that's honestly what those would play big roles in, like the cell would know based on the lipoprotein that you're supposed to take it from this component to this compartment to this compartment or something like that. They'd be essential for the transport of this particular bubble, right? But the bubble itself is really made of these glycerol proteins, these phospholipids, where they have the polar head, which is exposed to the polar outside environment, and then the non-polar tails, which are keeping all those non-polar lipids inside happy. All right, so here we go. Worst case scenario, not worst case scenario, but this is one of the things we're gonna have to be able to draw, is to draw the formula of a particular glycerol phospholipid. I'm not going to make us memorize the different names, But given these components, we're going to have to be able to use our constants page in order to figure it out. So this would contain two stearic acid fatty acids and a phosphate group, as they always do because it's a phospholipid, and serine, right? So this is all about using that constants page. So first of all, because we're in the glycerophospholipid section, I know that I'm going to have to start out, actually draw my carbons explicitly, with my glycerol backbone. For two of these carbons, I'm going to go ahead and draw the ester bond to the fatty acid. And the fatty acid that I'm specifically going to be included is this stearic acid. So now I got to go to my constants page, figure out what stearic acid is. Appears to be a saturated fatty acid with 18 carbons. So here's one here. Remember, don't forget to count that carbonyl carbon. I must have miscounted somewhere, right? Because 1, 2, 3, 4. See again, extra one. That's why we always double check by counting carbons. Let me even triple check. 1, 2, 3, 4. Okay. So this is now 2/3 of my glycerophospholipid here. Now I need to include the serine component, which is going to be held by a phosphoester bond. So let's just draw that. Importantly, don't forget one of those oxygens has a negative charge on it. And now I need to find my amino alcohol serine. So which one is that? It's going to be an oxygen, CH2, C with the amino group, and then actually the alcohol group as well. So this has both a amino group and a, an ammonium group and a carboxylate group. So 3 carbons. This last one was a carboxylic acid. And the second one contained an ammonium ion. Let me double check that that's correct. All right, looks good. So this would be the cephalin, this particular glycerophospholipid here. And again, I'm not expecting you to know that this would be cephalin just by off the bat, but if I told you to draw me a glycerophospholipid that contained 2 stearic acids and serine, given our constants page, I would expect you to be able to. Okay, well, let's see if we can classify the following as being either fatty acids, triacylglycerols, amino alcohols, or glycerophospholipids. So let's see. Glycerol trioleate. What would that one be? Okay, so remember that this would be the name of when we made our triacylglycerol. It would contain the glycerol refers to that glycerol backbone, and then the trioleate would let us know, we could even draw this one given our constants page here, knowing that it contains 3 components of oleic acid. So this would be a triacylglycerol. Cephalin, well, we kind of just did that one. And again, I'm not actually going to, that's not going to be on our list of things that I expect you to just have memorized, is the fact that cephalin is a glycerophospholipid, although we did just do that example problem. So hopefully that was fresh in your brain. Choline. Choline is actually on our list here of amino alcohols. And palmitic acid, that's also on our list, but over here on our list of fatty acids. All right, so now sphingomyelins, or also called sphingolipids. Again, myelin is a component of a nerve where these are very much so found all the time in these myelin sheaths. They are abundant in nerve cells, so they play a fairly specialized role. They have their own sort of special backbone here, right? This sphingosine. They also contain these two common components that we know, one fatty acid, and then this phosphoester bond to an amino alcohol. Okay. Again, so you can see how they get the name sphingomyelins because they're present in myelin sheaths, which is the long part of a nerve cell. They're also very present in white matter, which is in the brain. Again, kind of just a brain cell, kind of nerve cell kind of deal. They increase the speed of neuroimpulses as well as insulate and protect the nerve cells. And that protection is a fairly important role. If multiple sclerosis is a disease where sphingomyelins are lost from the myelin sheaths of nerve cells, and that leads to this deleterious kind of disease where in general motor function and movement is slowly impaired over time as these sheaths get worn. So that actual protection and insulation is really important, both probably from a physical protection standpoint, protecting it from being damaged, as well as just protecting the signal from being sent down those nerve cells efficiently. And this, okay, so now let's look. So again, we had these normal components, the fatty acid component, we've seen that before. The amino alcohol component held together by those phosphoester bonds, check. What's one of the weird things is that it's got its very own little backbone that includes part of a tail, and that's called the sphingosine. So you don't have to know the structure for this except for, being able to use it when it's given, but let's just take a look at it. So first of all, we can see one of the unique features is we don't form ester bonds with our fatty acid. What do we call these? When it's a carbonyl group, here, let me actually clean this up. What do we call a carbonyl carbon bound to a nitrogen? These are amide bonds. So right off the bat, we can see that we don't have any normal ester bonds. We have our phosphoester bond. But what would normally be an ester bond to a fatty acid is, in the case of our sphingomyelins, it's an amide bond. And then this other tail, notice that this portion is also a very long carbon tail, but it's now connected to this backbone, this sphingosine backbone. Okay, so this sphingosine is its own kind of thing. There are no normal old ester bonds in there. Okay, let's draw a condensed structural formula for a sphingomyelin that contains the 16 carbon saturated fatty acid palmitic acid and then the amino alcohol choline. Okay, so this is now, if we're going to draw one of our sphingomyelin structures here. So first of all, we're going to go to our constants page. We know that any sort of sphingomyelin is going to have the root of sphingosine. So I'm going to kind of be lazy and just go ahead and copy this guy here. Paste it over here. So this is going to be my root, unlike normally when I would have started with a glycerol backbone. Now I'm going to start with this sphingosine backbone. I'm going to get rid of these hydrogens. My fatty acid is connected through an amide bond. So palmitic acid, let's go back over here to our constants page. I have to find palmitic acid, which is a saturated acid, fatty acid that contains 16 carbons. Let's go ahead and put that in. The only difference being it's not going to be an ester bond. It's going to be a amide bond. So that's 1. So 1, 2, 3, 4. Let's go ahead and double check. 1, 2, 3, 4, 5, 6, 7, 8. Okay, and then the last component here is going to be the choline. So let's go over to our amino alcohols page and make sure we pick the proper one, choline. So we have 1, 2 carbons, and then a quaternary amine. So this would be the sphingomyelin of eggs, apparently. But anyway, importantly, we know how to build it because it told us which components from our constant page to use, right? The palmitic acid, the choline, and because it's a sphingomyelin, we know that we need to use that sphingosine kind of root there. Oh,